So my name is Ryan Giesecke. I'm president of Trinity Valley Beekeepers up in Dallas. I'm at the master level of the Texas Master Beekeeper program, pretty active in beekeeping around the state, working on stuff with TBA committees, as well as with the committee with the new Lone Star Beekeepers Association, and really just trying to have a positive impact wherever I can. In this case, we're talking about mites, and that's an easy spot to have a positive impact. Keep in mind, this presentation is going to be a bit of satire on the fights about mites, blame game portion. It's going to be a bit of serious information on the beginning portion where we're talking heavily about varroa mite biology as well. So without too much further ado, I'm going to go ahead and jump into this because it's a bit of a lengthy presentation and I'm sliding it in under the wire here today to make it fit into this block. So I'm a little bit of a big picture guy. We're going to start by talking about what mites are. Mites are arachnids, many species, decomposers, predatory mites, parasitic mites, live in a wide variety of habitats, upwards of 48,000 species of mites described. Just to give us some idea what an understatement or a broad generalization we're making when we say mites in our hives, right? We're usually talking about varroa mites, but even in beehives, there's upwards of 170 species of mites described, right? I'm going to breeze through these, but an example of another harmful mite would be the tracheal mites that have been a problem in the past for American beekeeping, as well as beekeeping in other parts of the world. Reproduce in bees' airways, not really a problem these days. Why? Because the bees have developed hygienic behavior that provided resistance to the tracheal mites, okay? There's also some neutral species of mites that live in a beehive. This is an example of one. Areoglyphus feeds on dead bees in the hive primarily. No real positive impact, no real negative impact. Another neutral to maybe slightly beneficial species of mite that lives in our beehives is mellitifus mites. And these feed on debris on the bottom board, specifically pollen debris on the bottom board. Slight positive impact perhaps, a little bit less mess in our hives attracting other problems. And there's even some examples of very clearly beneficial mites. Stratiolaelaps mites have been experimented with as a treatment against varroa mites. These are predatory mites. They can be introduced into a beehive. They will go crawling around on the comb. They will eat varroa mites that they find. Unfortunately, they tend to migrate out of the hive in favor of their natural environment, which is in the soil. So no amount of food sources in the hive is going to provide a sustainable situation in terms of preventing varroa mite problems. They're simply going to leave the hive as fast as we can put them there, which is why they haven't evolved into a treatment that's actually in use, okay? But today we're primarily talking about varroa mites, and with good reason. These mites have been major problems for modern beekeeping. They are well, they were originally discovered in Southeast Asia in 1904 as Varroa jacobsoni. We heard a little bit of this mentioned in the big room just a second ago. We're going to go into a little bit more detail. They were first found in the US in 1987. Basically, they feed on the bodily fluids of our bees and create problems for our bees, vector disease, other issues. Apis cerana, we talked about the western honeybee that we keep, right? Apis cerana is the eastern honeybee. It's very similar to the bee we keep, but a different species in the same genus. So Apis cerana is where these mites came from. They didn't just spontaneously come out of nowhere. But since they've spread onto the bees we keep, the western honeybee, Apis mellifera, they have really devastated beekeeping worldwide, with the notable exception of Australia. This slide says still not present in Australia. Some of you may have seen the news story as of yesterday that there's another big scare that they just got introduced to Australia. Past scares along those lines have been Varroa jacobsoni, which is the mite that still lives on Apis cerana, accidentally getting introduced to their continent. The scare from yesterday was instead of a ship coming from Asian origins, where it would have been Apis cerana bees accidentally brought in, yesterday's scare is bees accidentally brought into Australia from the United States. And so we may very well be right on the edge of seeing a major outbreak in Australia. Hopefully that's not the case, right? So you'll see two species names on that slide, and the reality is that there's 5 species of varroa mites. We don't have to worry about most of them because These bottom three have absolutely nothing to do with the bees we keep. Live on other species of bees, all right? Varroa jacobsoni is the species that lives on Apis cerana present day. And Varroa destructor is the species that we're worrying about in our hives. But these first came into the US in 1987, right? And if we look up at that slide, we see that Varroa destructor was described by science in 2000. Until that point, it was still not differentiated from Varroa jacobsoni. So I'm going to be referencing sources that mention both, because any source from before 2000 is going to say Varroa jacobsoni, even if it means Varroa destructor. Does that make sense to everybody? All right. So let's talk a little bit about Varroa life cycle. Don't worry if you can't read everything on this slide. It's just the only image I could find up with to go with this slide. We're going to go through it in a little more detail. We're going to start by looking at developmental stages of Varroa mites, right? We know this about our bees. There's no reason we shouldn't understand Varroa a little bit better as well. It better equips us to deal with problems, to decide how to deal with problems. On this slide, we have some very light-colored mites. The light-colored mites, be they male or female, spend that portion of their life cycle inside of a brood cell, a capped brood cell. That light coloration tells us that their exoskeleton has not fully developed yet. They will promptly die of dehydration outside of a capped brood cell in these stages, which for a female mite is a young stage in their life cycle. Later in life, they're equipped to come out of the cell. But for the male mites, they never do fully develop their exoskeleton, and that's okay if you're a male Varroa mite. You're going to mate in the cell, your job's going to be done, and their entire life cycle is taking place inside of a capped brood cell, okay? Now for our female mites, we have them develop this exoskeleton where they can crawl out of a brood cell as it uncaps, having fed on the developing pupa inside of a brood cell, and they crawl out as a mature daughter. And then past that, we have a developmental stage that we call a mother or a foundress mite. This is a little confusing. If we have a mature one here, then what is changing up here, right? And the answer is that a mature daughter climbing out of her cell has not had a phoretic stage where she is feeding on an adult bee crawling around in the hive. So we have phoretic and we have reproductive mites, and these are both mature female mites that we're talking about here, okay? Phoretic mites live on the bees. They're usually not visible because they're tucked between exoskeleton segments on the bottom of the bee's abdomen. Photos like this are popular online for phoretic mites because you can see what we're talking about. Reality is it's going to look a lot more like this. A visual hive inspection is not likely to show you whether you have phoretic mites, okay? Not seeing mites does not say they aren't there. Phoretic mites prefer young nurse bees inside of the hive. And mites that do not have this phoretic stage have a lower fertility. In an artificial lab environment, if a mature female is taken straight out of a just uncapped brood cell, artificially placed in an open cell next to it, she will reproduce, but not nearly as effectively as if she had a phoretic stage first, right? Basically, she's gathering supplies for reproduction while she's feeding on that bee. In a natural environment, they will not skip a phoretic stage. They will have the phoretic stage first and then proceed to reproduction. So let's talk about reproductive mites. Again, guess what? Usually not visible, tucked away inside capped brood cells inside the hive. Despite the popularity of this kind of picture, it's going to look more like this. And unless we're breaking open brood cells to look for them, again, a visual inspection doesn't tell us whether we have mites or not, right? Reproductive mites prefer drone brood over worker brood quite strongly, ratio of 9:1. We're going to talk about timing in a second. That has everything to do with the timing of the developmental stages of the bee. And for the same type of reason, they never invade queen cells. They've developed a tendency to be repelled by royal jelly, and they will never invade a queen cell, which makes sense because they can't reproduce there. We're going to talk about why in a second. So first, let's talk about this reproduction process. Foundress mite enters the brood cell just before capping, okay? She crawls into a brood cell with a very mature larva in it. This larva is about ready to proceed to pupal development. The bees are about to cap it. The foundress mite crawls into the cell, down to the bottom of the cell, buries herself in the remainder of the bee food in the bottom of the cell for that larvae. and sits there, sticks up an air tube so she can breathe, and waits. Now the bees cap the cell, and the larva finishes off the last scraps of the bee food in the bottom and releases this female mite. She's already mated when she was originally in a cell earlier in her life cycle. And so what does she do? She starts laying eggs. She lays an egg about every 30 hours. Much like our honeybees, this is haplodiploid reproduction, which is to say that an unfertilized egg becomes a male, a fertilized egg becomes a female, okay? So she can choose, and the first egg she lays will always be a male. She's going to set up a male first, then she's going to lay the rest of her eggs as females. She's accomplishing this because she is going to need these offspring to mate inside of the cell and then come out ready to go through the phoretic stage, et cetera, et cetera. But females have to reach maturity and mate to be viable, and this takes about 10.5 days from where the egg is laid. So we're noticing real quick, there's a timeline here, isn't there? Typical number of offspring from a worker cell in a 21-day cycle of development is between 1 and 2. Typical number of offspring from a drone cell, which has a longer development cycle, 24 days, is somewhere between 3 and 4. More reproduction in drone cells, thus they favor drone cells, okay? So one of the questions that originally got me interested in the research that led to this presentation was trying to figure out how long varroa mites live. You ask beekeepers around you, and people look at you kind of confused and wonder why you want to know. And the reality is, it's kind of a confusing question, the same way talking about the lifespan of our worker bees is kind of a confusing question. Somebody asks me how long worker bees live, I usually say, oh, 5-6 weeks during the growing season. And if I add that last part, I'm right? At least close. If I don't add that last part, then I'm wrong for a certain portion of the year because they live longer in the wintertime. And the varroa mites, unsurprisingly, as a parasite, follow a very similar pattern. So the phoretic period may last 4.5 to 11 days when brood is present in the hive. But in a period of broodlessness, the phoretic period of a varroa mite may last 5 to 6 months. All right? Consequently, female mites living when brood is present have an average life expectancy of 27 days. And in the absence of brood, they may live for months. Confusing answer to a slightly confusing question. Regardless of what stage in their life cycle we're looking at, they are in there transmitting viruses. And this is the main problem with varroa mites. We tend to look at varroa mites as the problem. You know, in terms of us dealing with it, it's a logical perspective, but in reality, the problem is the diseases vectored by the varroa mites is what's killing our bees. They're spreading diseases to the bees both while they're feeding on brood and also while they're feeding on mature bees during their phoretic period, right? Lots of viruses, most of which are transmitted primarily by varroa, okay? We're not going to go into detail about these because reality is they don't impact our management much more specifically than knowing they're out there, except for deformed wing virus. We can see this bee right here with scraggly wing on one side and looks like almost none on the other. It looks like even some malformed feet on this one. This is deformed wing virus. And this one matters to us because unlike varroa mites that we can't see on a visual inspection of our hive, if you look in a hive and you see a bunch of bees with deformed wings, guess what? You have a varroa mite problem even if you don't see it. Now, it's not necessarily good enough to just look for this. You may see a problem too late to do something about it if this is your approach to looking for it. But this is a symptom we can watch for in our hives. And the reality is it's one of the most problematic viruses in the United States, according to this 2013 survey, just below black queen cell virus, which most of us, if we're not in queen rearing, are not going to specifically encounter in a way that could be symptomatic for our management, right? The other thing we can look for in our management is parasitic mite syndrome, which you'll commonly see referred to as PMS. Not to be confused with PMS, of course. Parasitic mite syndrome, essentially we're looking for this sort of scattershot brood pattern, but we're looking for not only the scattershot brood that might be confused with a failing queen, but the scattershot brood with these kind of spaces between it. Spaces where brood was clearly started and has since died and been chewed down by bees trying to clean this cell out. And in this case, we can even see a varroa mite on some of them, right? We're looking for dead brood between capped brood, not just scattered capped brood that would indicate a failing queen. All right? So Parasitic mite syndrome, again, a symptom you can watch for, but by the time you see it, it may be too late to try and recover this colony, okay? So let's talk a little bit about how varroa spreads from colony to colony. And in this case, we're going to talk about how it spreads without our help, okay? So it spreads first off when colonies abscond or swarm. Obviously, we've got mites, varrotic mites on the bees. If our bees are traveling, guess what? Mites are traveling too. They may travel miles that way, right? And they're also traveling other ways. Worker drift, for example. A lot of us probably know that our bees know exactly where the entrance to their hive is, right? Has anybody ever moved the entrance over 12 inches and watched the bees get confused? It's kind of entertaining if you're fairly bored. But nonetheless, they know exactly where the entrance to their hive is. We may think they don't drift into other colonies, but this 1998 study at the bottom that I put probably a little bit too small clearly verified that workers from highly infested colonies have a greater tendency to drift into other colonies than workers from colonies that are not as infested by varroa mites. So worker drift. Drone movement, conceivably this plays some role as well. Drones are for the most part welcome in any hive they stumble in the door to. They're not a preferred phoretic host for our phoretic mites, but conceivably it plays a role. Another probably fairly minimal role would be flower hopping. This screenshot here is pulled from a video that you can find on YouTube if you look up flower hopping mites. It's both fascinating and fairly terrifying watching this mite fairly nimbly jump onto the next bee that comes by this flower if it gets abandoned on the flower. So this would be a way of transmitting from colony to colony. And robbing, of course, robbing of a collapsing colony plays a major role, but we're going to talk about that a little bit further in, okay? A study from, I believe it was France, and I forgot to reference it, showed that within about 3 months, mites spread into colonies about 4 miles away when this problem was first spreading across Europe, okay? Now, this is where we jump to the second part of the presentation, where this presentation gets its name. The Fights About Mites is the varroa blame game, okay? There's a bit of a satirical element to this presentation, all right? This is intended to educate us on bees and mites, but it's not actually intended to assign blame. There's only one way you cannot do any of the things that I blame for our varroa mite problem in this presentation. Anybody want to guess what it is? Don't be a beekeeper, okay? So if you're feeling picked on, just give me a minute. I'll pick on your neighbor next, I promise. But we're going to start, innocently enough, blaming Apis cerana. After all, this is where this mite problem originally came to our species of bees from. Apis cerana lives in South and Southeast Asia, very similar to our bees. Multiple layers of comb inside a cavity, managed by beekeepers in hives not dissimilar from ours inside of their native range. And basically not managed outside of that native range. Many similarities with the bees we keep. If you haven't seen a comparison shot before, here's a comparison shot. We can ignore the giant honeybee across the top. This is our bee in the middle, and Apis cerana across the bottom, okay? Now, how does Apis cerana deal with varroa, right? And why doesn't this work for our bees too? Well, there's a list of ways that they deal with varroa here. First off, self-grooming behavior. And grooming behavior we arguably see in some hygienic subspecies, subhygienic races, especially in selected breeding in the species of bees we keep. But the second one on the list here, dance communication, to my knowledge, there's no evidence of in our species of honeybees. Dance communication saying, hey, I'm doing a little dance because I've got a bee on my back. Somebody come help me with this, okay? Which of course leads to nestmate grooming behavior. Again, to some extent, maybe in some hygienic lines we have in our species in the United States, but probably not to the same degree that it is in Apis cerana. Mite chewing behavior. I'm not just going to groom this mite off, I'm going to disable it so it can't get back on another bee. There are some programs that have specifically selected for this in the US. Purdue has a program some of you might be familiar with. They mite biters, or I've heard them called ankle biters as well, because they tend to chew the legs off of the mites. It's a resistance trait that's being specifically selected for in our bees. Removal of infested, carrying mites out of the hive. Again, this is kind of general hygienics. We can't specifically say it doesn't ever happen with our bees, probably not to the extent as Apis cerana. Removal of infested worker brood. And this is not talking about just, hey, I'm cleaning out dead brood. This is talking about removal of brood, even if it's potentially still not quite dead, because there are mites in the cell. And I've got social apoptosis under it there. I kind of stole this from a Randy Oliver article some of you may have read in American Bee Journal. Apoptosis is what our body does when it sloughs less than perfect cells to keep problems from spreading to the rest of the organism, right? Social apoptosis would be when individuals are sacrificed inside of a social colony for the sake of keeping the colony as a whole strong. We see it not only in Serrana with varroa mites, we also see it with nosema in some cases with the bees we're keeping. Serrana has pores in the cappings of drone cells, which may enable them to monitor a little bit better what is happening in terms of mite reproduction inside of drone cells. And they tend to seal in infested drone brood. They're not even going to break that cell open and give the mites a chance to run off of it. They're just going to seal it in with propolis, deal with this once everything's dead inside, okay? Increased swarming and absconding rates probably play a significant role as well. This is one of the reasons Serrana isn't popular in management. But first and foremost probably is our last item here, which is shorter worker development. Remember, we said timeline matters here. We talked about the timeline for varroa mite reproduction. So what we see is a chart that I've added red numbers across the bottom of here showing how many mites are generally reproduced inside of these cells. And what's the key number here is the pupa to adult number, referenced in the study at the bottom as a post-capping time. Post-capping time with Apis cerana workers is 11, a little bit longer with our bees. Drones, of course, are longer in both species. And of course, if it takes 10.5 days inside of a cell, queens of either species are totally safe from varroa, which is why varroa don't favor queen cells. Does that make sense? All right. Oh, and the study at the bottom says 30 to 60% mite reduction just by having a 10% shorter post-capping time. 10% of 11 and 12 is somewhere right around a day, which is in fact the difference we see between the two species. And lots of people do blame Apis cerana. Clearly, Australia has worked hard to keep Apis cerana off of the continent for the sake of preventing the spread of varroa. And they're really just not popular outside of their native range to the extent of very organized extermination programs in some cases, despite the fact that they're arguably a better crop pollinator for some circumstances because they have a smaller forage range. And they are very self-sufficient and resistant to pest and disease problems. Now we're going to jump on to the next stage of our blame game, which is we're going to blame Russian beekeepers and the Trans-Siberian Railroad. Because after all, they had to get off of their bees somehow, Apis cerana somehow. And this happened when Russian beekeepers brought beehives containing the same species of bees we're keeping down the Trans-Siberian Railroad in the early 1900s and out into the Korean Peninsula. They set these bees down right next to where Apis cerana was being managed inside of their native range. Sure enough, these mites looked at the hive right across the fence, so to speak, and said, ooh, that looks like a good place to be too, right? The grass is always greener over there. And so they jumped onto the bees we're keeping. Basically, we can see some comparison of cell size here, which explains why there was a tendency not only to jump into the box next door, so to speak, but also a tendency to start going into worker cells significantly, because our worker cells are approximately the same size as Apis cerana drone cells. All right? If you wonder if those numbers really tell us how similar they are, there's also a study at the bottom here that says if I put Apis mellifera foundation, worker foundation into a cerana colony, they will use it to raise drones in. Very similar cell size. And then, of course, we, Russian beekeepers rather, brought those beehives back off of the Korean Peninsula and conceivably right back down the Trans-Siberian Railroad. And the spread of mites across Europe is frequently attributed to a military base outside of Berlin where those hives were set down. And basically, this is when we saw a differentiation between these two species of varroa mites, right? Now, that wasn't a big problem for beekeepers in the US until they got to the US. At that point, we probably have to start blaming American beekeepers a little bit, as well as possibly USDA and APHIS, although they were doing their best to keep this problem out of our country. Varroa mites came into Florida, presumably on imported bees, despite quarantines that were supposed to prevent this. They were first documented in hives that collapsed moving from Florida to Wisconsin in 1987. Early spot checks revealed them already in 10 states, right? Now, this bottom item, managed colonies dropped by almost half from 1945 to 1990. You frequently see this referenced in terms of look at what an impact varroa mites had. But we also have to acknowledge that this high point in the 1940s probably had a lot to do with World War II, not a lot to do with the fact that varroa mites weren't here yet specifically. This drop from 85 to 90 is clearly indicative of the varroa impacts. All right? Now, Florida to Wisconsin. Florida and Wisconsin are not neighbors, right? So the reality is migratory beekeeping played a role in spreading this problem as well. If Florida and Wisconsin isn't enough of an example, I know you probably can't read these numbers at the bottom. Suffice it to say the darker states got varroa mites first and the lighter states got them last. We definitely don't see an orderly march of mites across the continent, right? So they're jumping from state to state, moving faster than 4 miles in 3 months. Migratory beekeeping is playing a role in this. This despite the fact that inspections of migratory operations do intend to catch things like this, reality is you can never hope to inspect every hive with the number of hives that move around the country. You inspect, spot check, and you hope you're catching things. Honey production also plays a role in the blame game here. This study from 1944, by the way, has anybody ever stacked boxes this high? I haven't either. I don't know why you would. It's just a reason to break out a ladder at that point. But this study from 1944 shows us that hives with a stronger population make more honey. Not only more honey in the sense that it matches the number of workers, they actually make more honey per worker as a hive gets stronger. So there's every reason in honey production to get strong hives and keep strong hives, right? Unfortunately, Tom Seeley's studies on hive populations and the way feral bees combat varroa say that smaller hives deal with varroa problems better. So by keeping strong production hives, we're unfortunately having a negative impact on our mite situation. Of course, if we're blaming this ever-expanding stack of boxes, we could probably go ahead and blame Reverend Langstroth as well. But since a lot of us are using equipment inspired from him, let's not go too heavy on that one. We are going to talk about hive proximity and bee yard density. Naturally occurring feral or wild hives are widely spaced. They don't tend to pile lots and lots of colonies into one location. Numerous hives in close quarters increases risk of mite spread from one hive to another, right? You just jump through that wall and find a new host. And in fact, this proximity leads to increased virulence in our mites. It's basically the same kind of situation that humans saw in Europe with the plagues when we started seeing how many people we could pack into cities, right? Pests and diseases, parasites and diseases tend to not kill their host if there's not another host available, right? Another host always available a few inches away leads to parasites that kill their host, okay? If you doubt my word for it and my analogies for it, study from Nolan and Delaplane in 2016 says colonies spaced only 100 meters apart have much better mite counts than 0 or 10 meter apiaries. Now that said, does anybody here have more than one hive and have 100 meters between each one? I've never gotten a yes before. Did you say yes? That's awesome. That's awesome. Congratulations. So most of us don't do that because beekeeping admittedly is a balance between what the beekeeper needs and what the bees need, right? I always tell people, you know, the bees would rather be 20, 30 feet that way. But none of us are doing that because this is a compromise, and hopefully our management has enough benefit to outweigh any downside we've created. Now, it probably looks like I've been picking on professional beekeepers a little bit. That's not my intention. So for our next slide, we're going to look at some numbers from TIIS. These were reported in 2017, and these are mite counts from their inspections of beekeepers in Texas. Commercial beekeepers came in at 1.5 mites per 100. Sideliners, which would be people that are depending a little bit on beekeeping for an income, but not primarily on beekeeping for an income, came in at 1.8 mites per 100. And the hobbyists came in at almost 5 mites per 100 bees. Looking strong, right? Now, these numbers may be a little bit biased by the fact that commercial beekeepers tend to call the apiary inspection service when they know they have healthy hives and want to get them to California. And hobbyists probably tend to call the apiary inspection service when their colony is dying or even dead. So that may play a role in this, but the reality is something's being done to combat mites over here, and we can't say it's not working, right? Now, probably the bulk of this blame game topic comes from this fight between treatment-free beekeepers and, let's call them, treatment-inclined beekeepers, okay? And this is not without reason. Individuals reporting, we're going to pick on treatment-free beekeepers first. And when varroa first hit the country, individuals were widely, almost unanimously reporting that any colony not chemically treated for varroa mites was collapsing under varroa mites. If you want your bees to live, you had to treat them, right? Feral populations had not disappeared, but all but disappeared by 1995. And even in a more recent study from 2009, we see varroa mites are associated with 85% of colony deaths. That's a study from Canada. And of course, we already talked about hives in close proximity collapsing are leading to not only more and more varroa, but more virulent varroa. So if you're not treating for varroa mites, hives are collapsing, leading to more virulent mites and spreading elsewhere, right? Perhaps the main reason we see treatment-free beekeepers blamed for everybody else's varroa mite problems is because of mite bombs. And this is where I told you we'd get back to talking about robbing, okay? Mite bombs, for those who aren't familiar, happen when a healthy hive has a reasonable mite count and then the mite count starts to rise, okay? Now, as the mite count gets worse, the colony starts to suffer, and the population starts to shrink. Population's shrinking as mites reproduce, so the mites per bee is getting higher and higher. As it gets to a critical point, the colony starts to collapse under the mite load. And at this point, it starts to get robbed of its honey by surrounding colonies coming and stealing what they can, right? They're carrying mites as well as honey back to the surrounding colonies. Meanwhile, this colony is doing so poorly that it goes queenless. At the point when it goes queenless, the colony continues to get robbed, and now these queenless workers have a tendency to start to drift off into surrounding colonies, literally following the robbers back to these other hives that are doing well at the moment, right? Before long, our original colony is completely dead, and surrounding colonies are likely to be left with much higher mite loads, okay? This is a reason to blame somebody else for your mite problem, all right? That said, we need to be realistic about mite bombs. The reality is all colonies do have mites, okay? It's not a matter of do you have them, it's a matter of how many do you have. Any colony that collapses has the potential to create a mite bomb. Maybe varying degrees of how bad a bomb, nonetheless a mite bomb. Hive density in an area has a major impact. If one colony collapses and there's 3 colonies within range of robbing it, that's a heavy mite load into each of them. If there's 100 colonies within range to rob it, hey, that spreads the mites out a little bit, right? But the reality is, assuming colony loss rates in your area are anything above 0%, then any mite management plan that can't weather mite bombs is ultimately doomed to failure, okay? So treatment or non-treatment is really irrelevant. The question is your management plan capable of dealing with the inevitable mite bombs? If it's not you or your neighbor's hive collapsing, maybe it's a feral colony, somebody's going to collapse at some point. But we've blamed treatment-free beekeepers, and we're going to blame people that are treating as well. You know, studies in Africa show that untreated colonies developed resistance in Africa. This was largely an economic decision that led to a lack of chemical treatments in African beekeeping. There also tend to be a little bit more hands-off beekeeping than we are, partially because bees are wildlife there rather than as domesticated as we tend to view them. Tom Seeley's studies show that a genetic bottleneck from die-off ultimately led to resistant genetics. These were studies done in the US, up near Ithaca, New York. Major die-off led to a small survival rate that reproduced and produced bees that were more capable of dealing with varroa mite problems. And we get to blame our neighbor for our resistant stock not working as well because of how honeybees mate, right? Propping up non-resistant bees with treatments can be said to flood drone congregation areas where our queen's going to mate with non-resistant genetics. If my queen takes her mating flight and comes back less resistant than her predecessor, I can tell my neighbor it's his fault because he's been treating his bees. So we see this is not without reason that this is a fight, right? We can literally place the blame. You see sometimes the chemical treadmill reference, and I have to admit I somewhat stole this graphic from Liz, but I did find it on Google before I stole it. The chemical treadmill is the argument that ultimately using chemicals to treat varroa mites is going to lead to more and more resistant varroa mites that we can't treat without using harsher and harsher chemicals. I think you could even go as far as to talk about the treatment treadmill, even without chemicals in the picture. We could talk about things like culling drone brood as potentially on a long enough timeline leading to bees that favor workers, right? We could talk about treatments like oxalic acid that don't penetrate capped brood cells as potentially on a long enough timeline developing mites that need less and less phoretic period, more and more time spent sheltered inside of a brood cell. No real scientific evidence of that, but it sort of makes sense if you think about it. As we talk about resistance, there's several things I want to consider. First off, we can talk about resistance in its real scientific definition, which is the ability of a host to limit a parasite burden. Okay? This is, again, somewhat stolen from Randy Oliver. We could also talk about tolerance in terms of an effort to survive, the ability of a host to limit the damage caused, but not the parasite burden. Now, our mite counts can definitely check for resistance. Our mite counts don't so much check for tolerance, do they? Only survival checks for tolerance. We could even talk about intolerance, which is back to that social apoptosis thing, right? An absolute intolerance, even if it's not leading to individual survival, may lead to colony survival, okay? Does that make sense? Now we're going to jump back into our blame game, and for the sake of expediency here, we're going to blame both swarming and not swarming on this colony, on this slide. Swarming and swarm prevention, if you will. Swarming is said to create a brood break which interrupts mite reproduction cycle in a parent colony, right? It's supposed to be a relatively effective way of reducing our mite population. Randy Oliver credits swarming with 60% mite reduction in the parent colony. But the reality is a lot of those phoretic mites simply get carried away with the bees that leave, okay? In terms of the brood break, mite reproduction can outrun bee reproduction. If it couldn't, we wouldn't have a problem with them to begin with, right? So if you really look at the fact that mites can live longer than bees, reproduce faster than bees, swarming is only truly beneficial to the overall bee population. We're worried about our neighboring hives too, right? If one, treatments are implemented during broodlessness to kill off phoretic mites, or if two, hygienic grooming behavior is so strong that it has a similar impact during broodlessness. Because the reality is, if mites are simply getting carried away from our colony, where are they going? Anybody?
The next colony.
To the next colony, right. And unless our neighbor happens to be out there catching all of our swarms and treating them, they're likely to be carried off into the feral honeybee population, especially in the south, okay? And many beekeepers do believe that the modern feral population contributes to this problem and is to blame for part of the issue. Many people believe that the feral population simply consists of short-lived colonies that are constantly dying, constantly swarming, and constantly producing mite bombs, and just outrunning the mites just long enough to swarm once and then die off. You know, I work in bee removal, and sometimes I get a call from a bee homeowner who says, hey, I've had this colony there for 10 years, but the reality is we don't know. They could be dying every fall and new colony moving in every spring, and they would never know the difference, and I wouldn't either. We can blame foundation. After all, cell size is often a factor in discussions in some audiences and some crowds. Standard foundation is larger than natural cell size in the vast majority of situations, producing larger bees. Standard foundation is probably more aptly named large cell foundation, and small cell is closer to natural foundation. Lots of arguments about this. We have a couple studies here at the top that imply that small cells tend to help. Then we have a study right below that that says there's no correlation between cell size and varroa mite problems at all. Study below that says actually small cells are far worse. Study below that says, oh, well, but if we just raise workers in drone cells, then that solves the problem. Extra large cells are good. So let's ignore all that for a second, and let's point out that brood developing in larger cells, larger bees developing in cells, need more visits from nurse bees to feed them more food in the development cycle. So more visits from nurse bees with phoretic mites on the bottom of their abdomen may lead to more introduction of mites into the cells. And some historical observations from before foundation seem to imply that natural comb was better, in the sense that natural comb had shorter development times. So present day numbers tend to say something around 21 days after an egg is laid, we get a mature worker bee. 1792 reference said 18 to 20 days after an egg is laid. and an 1882 reference, just shortly before the popularization of foundation, said 19 days after an egg is laid, we get an adult worker. Now that should seem a little bit familiar from somewhere, because that's exactly the number that Apis cerana is using to be able to resist varroa in its worker cells, okay? Now you've noticed a pattern here, so we're going to have to blame foundationless beekeeping as well. Reality is foundationless beekeeping tends to produce more drones. A feral colony of bees builds about 17% drone comb. Tom Seeley told us that. Randy Oliver tells us that if we regularly cull old combs and use worker-sized foundation, we can keep down about 44% drone cells in our colonies. 4% versus 17, that's a big difference in how many varroa mites may be reproducing in drone cells, right? But this only works if we regularly cull old combs, says Randy Oliver. So even if I change this title and we talk about not culling old combs, we have another angle to blame, right? Not culling old comb could contribute to the problem. Foundress mites prefer to invade cells in old brood comb. Compare 22.6% of cells invaded in old comb to just under 10 in fresh new comb when you drop both into a colony. Now the reality is all those mites were going to go somewhere regardless. Nonetheless, strong preference for old comb. But if we don't cull old comb, there's potentially an advantage to that as well, believe it or not. And this is shown from a study from 2012. Mites invading brood cells in old comb have fewer offsprings, as does each foundress when multiple mites invade a cell. This is because varroa mites produce pheromones, and they produce a pheromone to inhibit competition inside of a cell to try to favor their own offspring. So this Z8 heptadecene inhibits varroa mite reproduction, okay? So this builds up in old brood comb and conceivably starts to inhibit varroa mite reproduction. In a lab, it was shown to lead to a 30% reduction in mite reproduction in cells. Now, unfortunately, this did not translate well into field trials, and it has not become a viable treatment for us. Again, not that different from the stratiolalaps, right? Urban beekeeping and keeping calm bees. It's great to have gentle bees. Anybody really like it when your bees try to kill you? Okay, no. Nonetheless, a lot of the feistier genetics, notably the African and the Russian genetics, show some increased resistance to varroa mites. African genetics show lower levels of infestation as well as higher percentage of infertile mites in the mites that are produced in their colonies. Compare 100% infertile in Apis cerana worker brood to 40% infertile in African stock, to 10 to 20% in the average American colonies, with the Italians that we favor as gentle being right around that 10% mark for the most part. So as much as we like gentle bees, less gentle stock is considered manageable in a lot of the world and tends to be more varroa resistant. We could also blame genetic diversity or a lack thereof for part of our population, part of our problem with this. Feral populations that develop resistance through an extreme genetic bottleneck, as described by Tom Seeley, have to start off with genetic diversity in order to do so, okay? If you have all the same genetics in every hive, like we tend to get with grafting, vast numbers of queens produced from a very limited number of parent hives, this can lead to genetic diversity problems. Sue Kobe's team at Washington State has been importing frozen bee semen to use in instrumental insemination projects to address specifically this problem. And a lot of these bees that are reproduced this way are Italian stock, Apis mellifera ligustica, and they tend to be especially susceptible to varroa problems for specifically one of the main reasons we like them, and it's not that they're docile. Part of what American beekeepers, commercial beekeepers, like about this stock is that it is able to brood up big in the wintertime so you can make grade to get to almonds, right? Sounds like a good thing. It's a good thing for nectar flow. It's a good thing for pollination contracts. But in the average temperate climate, mite populations can increase 12-fold in colonies having brood for half the year and 800-fold in colonies having brood all year round. So raising brood through the winter has a problematic impact on our varroa mite populations. All right? Now, what am I recommending in all this? I'm recommending that we not treat blindly and prophylactically, that we treat because we have a problem, standard IPM approaches, and I think there's going to be some other talks on that today. I'm recommending that we avoid mite bombs whenever possible. Even Tom Seeley in his Darwinian beekeeping article says, hey, don't create mite bombs. If a colony is collapsing under mite load, kill it. Close it up. Don't let those mites spread. Don't increase virulence in our mite population. No reason to watch it suffer. If it's done, do it. Okay? And lastly, just be aware of your mites. There's nothing I'm more tired of hearing than, well, I did a hive inspection and I didn't see any mites. That's not the answer. It's not an approach that's going to tell you anything. It's not going to solve anything. Just like blaming somebody else for your problem isn't going to solve anything. So we've blamed a lot of people and a lot of things for our mite problems today. Hopefully this slight satirical element has encouraged you not to blame your neighbor for your mite problems, and instead to make an intelligent, informed decision about what you are going to do about your mite problems on your bees. So maybe we can spend a little more time intelligently discussing the situation and a little less time fighting about mites. Thank you guys for listening.